Hello everyone, this is Kunika Chandra from Shivaji College, Delhi University. So in this video, we will be continuing our previous topic on amino acids. So let's have a quick overview at what this lecture would hold. So the first part of the video was where we covered some basics of amino acids and how they're prepared and some reactions, also called the chemical properties of the amino acids. In the second part, we would be starting with the physical properties of the amino acids where we'll be discussing their isoelectric point and their Zwitter ionic form. Next, we'll move on to a small distinguishing test called the Nonhydrogen test, and in the end, we'll do a small separation technique called electrophoresis, which we'll be using to separate a mixture of alpha amino acids. So a small test that all alpha amino acids show is called the Nonhydrogen test. In this, we can see that all amino acids having alpha amino group would give a violet color dye with Nonhydrogen. So Nonhydrogen is a reagent. If the amino acid is not present in a solution, it would leave the solution as it is, also called the negative Nonhydrogen test. And if the amino acid would have the alpha amino group, then it would be a positive Nonhydrogen test, giving a purple colored complex. But if the amino acid is a proline, which is an exceptional case, the Nonhydrogen would give a yellow colored compound. Also, the intensity of the color would increase when the concentration of amino acid is more in a solution. In the next slide, we'll see the mechanism of this Nonhydrogen test. So first of all, we've taken an alpha amino acid and reacted it with a mole of the Nonhydrogen reagent. We'll get carbon dioxide, aldehyde, ammonia, and the reduced form of the Nonhydrogen. In the second step, we have taken the same reduced form of the Nonhydrogen along with the ammonia which was expelled in the first step and reacted it with another mole of Nonhydrogen to get the violet-blue condensation product in a positive Nonhydrogen test. Moving on to the physical properties of the amino acids, the first one is the Zwitter ionic form. Now, Zwitter ion is a molecule having separate positive and negative charge on the same molecule. Now, when we take an alpha amino acid, we'll see that it is made of NH2, which is a basic group. Now we know that a base actually accepts a proton to get protonated, and we get the protonated amino group. Similarly, the acidic group, which is the COOH, loses a proton to make the COO-, also called the carboxylate anion. Now, since an amino acid is having the both basic and acidic group in the same molecule, that's why they're also termed as amphoteric in nature. Moving on to the different ways amino acid can exist. So as we did the Zwitter ionic form, it exists only in the crystalline state of the amino acid. But if we take an aqueous solution of an amino acid, we'll see that instead of just the Zwitter ionic form, it actually exists as an equilibrium between the cationic, dipolar, also called the Zwitter ionic form, and the anionic form of the amino acid. Now, if we have a solution of amino acid and we don't know which form of amino acid is existing in it, we'll simply measure the pH of the solution. So if the pH comes out to be very low, that is acidic pH, the cationic form of the amino acid would be predominant. If it's neutral, we'll have the Zwitter ionic form, and if the pH is high, that is a basic solution, so the predominant form of the amino acid would be the anionic form. Let's move on to the second physical property that amino acids show, which is the isoelectric point. Now in this, if we take a titration apparatus and in the burette we fill NaOH, and in the conical flask we take a solution of amino acid and we don't know which form of amino acid, like cationic, anionic, or dipolar form is it. It's all in equilibrium. So when we start titrating it, a point will come when all the structures in this amino acid solution will exist as Zwitter ions or the dipolar form. So the pH at this point is called as the isoelectric point. So in this slide, we'll have a thorough understanding at how amino acids migrate under electric influence. So for this, we'll be taking two electrodes, cathode and anode, and place a solution of amino acid under them. Now we don't know that if the solution is acidic, basic or neutral, or which form of amino acid is predominant in the solution. So we'll simply place the solution under electric influence and see the reaction. So if the solution is acidic, as we did in the previous slides, the cationic form of the amino acid is predominant in the lower pH, and so it would get attracted to the cathode because negatively charged electrode attract cations. Similarly, if the solution was basic in nature, the anionic form of the amino acid would be predominant and would therefore get attracted to anode. But if the solution was neutral, so the zwitterionic form would be there and it would not migrate to any of the electrode. So this is called the cathodic migration, and this is the anodic migration. And pH at isoelectric point means a point where all the zwitter ions are existing. This would be calculated by the formula which is given below. Moving on to the final topic of this lecture, which is the separation of amino acids. So amino acids are separated through a method called electrophoresis, which is actually based on pH control and the electric charge present on an amino acid. So it is a really simple method to separate a mixture of amino acids. This is electrophoresis apparatus we have taken. In it, there are two electrodes, anode and cathode, and we have dipped a paper strip, which is the chromatographic paper made up of cellulose acetate in it, and in the center, we have placed a sample spot, which is actually the mixture of amino acids, and these two solutions we have saturated with a buffer solution to maintain the pH. Now, when we pass electric current through it, as we discussed in the previous slide, how on applying electric current, the various forms of amino acids like cationic or anionic or the zwitterionic form would get separated. So we'll take the same principle in this method and use it over here. So in the next slide, we'll discuss an example of how to separate a mixture of amino acids. So here we have taken a mixture of three amino acids, lysine, alanine and glutamic acid. Let's take the electrophoresis apparatus. So in the sample spot, we have taken a mixture of these three amino acids. So now on applying electric current, we'll see that the glutamic acid is acidic in nature because the COH get converted to the carboxylate anion, and we know that the anions would get attracted to the anode. Similarly, if we take lysine, which is basic in nature, it would be protonated to form a cation and hence would get attracted at the cathode part of it, hence separating it from the solution. And coming on to alanine, which has no net charge, it would not move at all to any of the electrode. Now, we can also understand which amino acid would move to which electrode on the basis of the isoelectric point as well. So if the isoelectric point of the amino acid is lesser than the buffer pH, here we have taken the buffer as 6, the pH is 6. So if the pH of the buffer is greater than the isoelectric point of the amino acid, it means it would get migrated towards anode. And similarly, if the isoelectric point is greater than the buffer, which is in case of lysine, it would get attracted to cathode. So this is how we use electrophoresis to separate a mixture of amino acids. So that ends the second part of our lecture on amino acids. Let's take a quick recap at what we covered. The first thing we did was a small distinguishing test called the Nonhydrogen Test, in which we see that how to test a solution if it has an alpha amino acid or not. The second were some physical properties like the Zwitter ionic form and the isoelectric point. And in the end, we did a separation technique to separate a mixture of amino acids, which is called electrophoresis. So I hope this lecture was clear, and thank you everyone for joining.